Okay. Hi, everyone. Welcome to Quantitative Seminar. I'm Zoe. I'm an organizer this year, and I'm going to get us started with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes in the Suquamish, Tulalip, and Muckleshoot nations. Our lab also acknowledges that we live and work on the ancestral lands of the Duwamish people, the first people of Seattle. Now I'm really excited to introduce our speaker today. Celia Symons is an assistant professor at the University of California, Irvine. Her research focuses on the effects of climate change and other human impacts on lakes, with a recent emphasis on how global change drivers alter species interactions. She did her undergraduate and master's degrees at Queen's University under Dr. Shelley Arnott's supervision, where she researched the functioning of tundra ponds and their response to salinization. She then earned her PhD at UC San Diego, working with Dr. Jonathan Shurin, where she started studying lakes and ponds of the Sierra Nevada. Before she started at UC Irvine, Celia was a University of California president's postdoctoral fellow at UC Santa Cruz. She has continued working in the Sierra Nevada, combining observational and experimental work with synthesis projects to scale up to larger spatiotemporal scales. Celia would like people to hold their questions until the end. And so when that time comes, online folks, feel free to submit to the Q&A, and in-person folks, feel free to come up here to ask your question. And thank you so much, Celia. Take it away.
All right. Well, thanks so much for the introduction, and thanks everyone for coming to the talk. I'm excited to be here virtually with you and talk about some of my recent projects. I will be presenting some work looking at how species interactions and population stability vary with the environment. So before I get into the talk, I thought I'd start with some acknowledgements and a bit of a history on how this project came to be. So both because I want to highlight the people who are involved, but also because this program, Eco Days, is an opportunity that's available for people who are about to finish their PhD in aquatic sciences, or people who have just finished their PhD. So if there's anyone in the audience who's in that category, I highly recommend this opportunity. This is a program funded by NSF and ASLO and run out of the University of Hawaii. And Eco Days, what you do is you propose a project that you're interested in doing. So some sort of synthesis or conceptual piece typically that you're interested in. Then you get to go to Hawaii with about 30 other early career scientists, and you pitch your project, you join each other's groups, and you write collaborative papers. So all of the work that I'm going to present today actually began at Eco Days in 2018. And the project that I pitched for people to join at Eco Days was a project looking at the top-down and bottom-up effects of zooplankton and phytoplankton. I'm a freshwater ecologist. I'm interested in these dynamics generally, and my project that I pitched was looking at how species interactions change through time and with the environment. So I wrote a proposal using some kind of basic ways of looking at time-varying interactions. And I made this pitch and said the kind of data I had, which was time series of plankton in a number of lakes throughout the globe. And immediately after this, I had two people very interested in the project. And those two people were Tanya Rogers, who at the time was working with Steve Munch, and Alfredo Giron, who at the time was working with George Sugihara. So if you know those two names, you might guess that these two people came down very excited to talk to me about empiric dynamic modeling, a technique that they were both using in their postdoc and PhD work. So although it was a different quantitative framework than I proposed, it was kind of the perfect joining of forces through this chance meeting that allowed us to do all the work that I'm going to present today. The two main collaborators in this work are Tanya Rogers, who is now a research ecologist at the Southwest Fisheries Science Center, and Steve Munch, who is also at the same location as the program lead for ecosystem forecasting. This work is also based on publicly available data, long-term monitoring data that has been collected by many people painstakingly over many decades. And so I also want to acknowledge the huge contribution of those data sets and their availability to this kind of work. And I'll be noting those specifically throughout the talk. So jumping into it, part of my talk today will be addressing one of the oldest questions in ecology. What controls the distribution of biomass within and among ecosystems? There's some areas like the forest on your top left, where there are a ton of plant life, and others like the desert where they're more depauperate. There's also a lot of variation in the relative biomass of different trophic levels. So for example, famously, the African savannas support a very large biomass of animals relative to the plants, compared to places like the temperate forest. So I'm generally interested in determining what causes this natural variation. So let's zoom into a specific example. So here I have a kelp forest where we can see the kelp and fishes, but there's all the other marine life that you cannot see in the picture. So the question is, what determines their abundances? At the most basic level, the resources that are available in the ecosystem can determine the biomass through bottom-up control or resource control, which I'll be using interchangeably here, where the amount of primary productivity determines how much biomass can accrue at higher trophic levels. On the other side, there's also consumers or predators in these systems that can also control biomass through cascading top-down or consumer effects through their prey all the way down to primary producers. The balance of these top-down and bottom-up pathways in food webs are important to understand because the relative importance of these is likely dependent on the environment. So as humans shift environmental conditions, not only are we directly influencing the survival of species, but we're also altering the strength of species interactions, which can reshape ecosystems even in the absence of altered species composition. There's a ton of strong evidence that both resources and consumers can have tremendous impacts on ecosystems. And as a freshwater ecologist, here I've included two iconic pictures from whole lake experiments. So in these experiments, the lake is divided in half with an impermeable curtain. And in the top example, you can see the effect of top-down control from a trophic cascade from bass. When bass are removed, there is an increase in planktivorous fish, which decreases the abundance of herbivorous zooplankton and therefore increases the concentration of algae. So you don't need statistics to see that this shift causes a really large rearrangement of the ecosystem. So similarly, there's strong evidence that bottom-up control can also really have large impacts on lakes, as shown by the addition of phosphorus resulting in algal blooms on the bottom left. Now, although these are iconic examples of how important consumers and resources can be, the story that they tell kind of misses some other experiments where similar manipulations have been done, where the effects are much weaker or sometimes even have no effect at all. So how could that be? So there's a lot of different hypotheses, but one explanation is that the environmental conditions in these systems determine the strength of these species interactions. So zooming in on our study system of zooplankton as consumers and phytoplankton as resources, the bottom-up and top-down interactions between these groups might be variable over space and time. So these interaction strengths between the two can vary with the environment. One prediction is that top-down control will increase with warming, climate warming, because metabolism is more temperature sensitive than photosynthesis. The other thing that might influence the relative importance of these interactions or relative strength of these interactions is species traits. So for example, phytoplankton can grow spines or grow clonally in ways that make it harder for zooplankton to graze on them. So that can influence the impact they have on zooplankton, so this bottom-up effect, and zooplankton would then have a weaker top-down effect on these phytoplankton as well. In a similar way, species diversity in the whole community could influence interaction strength, largely via complementarity. So from these experiments, there are lots of insights about how the environment influences trophic interaction strength. But these experiments are often very limited in spatial temporal scope. The manipulations are rather extreme. So for example, here they are removing an entire trophic level, right? And so it's more likely in food webs that they're going to be affected by variation in abundance through time, not the presence and absence of a whole trophic level. The other thing is that experiments aren't always tractable if we're talking about really large ecosystems or ecosystems where maybe there's some ethical considerations about if we can do such experimentation. So one of our goals is to make better use of observational data to gain insight about species interactions. Monitoring data is collected over much larger areas, longer time periods, and capture kind of the messiness of nature. And increasingly, they are available for us to use, right? So there's things like the LTERs, Cal Coffee, all these time series data that are increasingly publicly available. So coming back to this initial setup, we wanted to see how the environment influences consumer and resource control so that we can predict something about how food web structure might change into the future. Okay, so in the past, one way to look at the relative importance of resource and consumer control in an ecosystem was simply a regression between the resource and consumer abundance. If they're positively correlated, this is assumed to be indicative of resource control, as when you have more resources, you can have more consumers, so a bottom-up effect of those resources on those consumers. And conversely, when you have a negative relationship, this would indicate consumer control, where more consumers result in a reduction of the lower trophic level abundance, so a top-down effect. One example of this kind of analysis that is fairly common in the literature is a paper here in 2015 looking at the sign of correlations between many different resource and consumer pairs in marine ecosystems. These relationships vary from positive slopes, so resource control in red, to negative slopes or consumer control in blue. So as you can see, there's a ton of spatial variation in these relationships. And in North America, at least, it looks like there's more resource control in lower latitudes and more consumer control at higher latitudes. So it tells you something about what factors are altering the biomass of these ecosystems. So this is a useful place to start. But as you can imagine, each location is only giving you one correlation value. So it doesn't tell you anything about temporal variation. It doesn't tell you about the relative importance of resource and consumer control, and certainly not if they're changing over time, which is what we were interested in. So as I said before, the approach that we took to accomplish these goals is to use a method called empiric dynamic modeling, or EDM. And EDM is on the far end of the continuum of models that range from highly mechanistic models where the relationship between each constituent part, so here species, is known and included explicitly in the model. The other end of that continuum is these equation-free methods, of which EDM is a part. And so historically, with complex systems, it was thought that we needed these mechanistic understandings, and we needed to painstakingly build it up, bottom up, by measuring the interactions of each component, each species. And if we're interested in environmental context dependence, not only do we need to know something about their interactions, but we need to know how they vary with the environment. So these approaches are hard to nearly impossible to scale up to natural ecosystems with hundreds of species. And so I think increasingly there's a movement to these equation-free methods where we can use time series data and the information contained in these time series to understand how complex systems like a food web behave and predict how they'll behave in the future. So what can EDM do that we can't do with the correlative approach that I talked about in the beginning? So here's an example with data on resource abundance and consumer growth rate on the y-axis. So you might think, well, growth rate of the consumer increases with resource. So this looks like a bottom-up effect with a lot of noise. So resource control with a lot of noise. But when you actually connect the points through time, you can see it's a little more complicated than that. So this is actually a complex predator-prey cycle that comes from a deterministic food chain model. And what you can see is that the relationship between resources and consumer growth could be positive, negative, or zero, depending on where you are and what trajectory you're on. And this, what EDM can do is then quantify the relationship at each time point. And it does that using a locally weighted linear regression or other non-parametric method, where the local slope coefficients tell you something about the interaction strength between resources and consumers. So importantly, these slopes are determined not just by what has happened most recently in time, plus random noise, like some models do, but by trajectories that are all the other times when abundances were similar. So for example, in this region where the slope is negative, we're using information from all the other times where the resource abundance and consumer growth were in this region. We can resolve cross trajectories by including axes for additional variables or time lags of existing variables in the model. So again, these methods let us look through time and see periods where we have resource or consumer control instead of just quantifying one metric for an entire time series. So using these methods, the purpose of our study, the first study I'll talk about, was to determine how bottom-up and top-down interactions vary through time, if there are any environmental drivers associated with this variation, and if those relevant drivers differ when we're looking across a bunch of really different lakes, or how they relate to variation within a lake through time. So as I mentioned, the focal ecosystem that we're going to be using are lake ecosystems, where we have zooplankton and phytoplankton abundance and environmental variables measured monthly for at least 6 years. This is, again, where I want to mention the importance of these long-term sampling programs. And certainly these kinds of analyses would not be possible without the data being shared widely. The data mostly come from the North Temperate Lakes LTER in Wisconsin, the St. Johns River Water Management District in Florida, and then individual lake sampling programs in Europe, Japan, and New Zealand. We chose these sites because EDM does not deal with a lot of missing data very well, and all of these sites have sampling that goes throughout the winter at monthly timescales, and it has been collected for many, many years. So those were important considerations when we were trying to find data. So I'm also especially appreciative that these sites are sampled throughout the winter seasons as well. So we collated data from 12 different lakes around the world, and these lakes span a wide range of nutrient conditions. So this plot here shows the trophic state index, which is basically a measure of greenness. It combines chlorophyll, nitrogen, and phosphorus into this index and tells us whether a lake is a lagotrophic, like Lake Taupo in New Zealand, that is quite clearly blue from Google Maps, up to eutrophic, like the lakes in Florida. We have Lake Beauclair pictured there, which is quite green. The interactions that we're interested in were the bottom-up effects of phytoplankton on herbivorous zooplankton and the top-down effects of herbivorous zooplankton on phytoplankton. We used chlorophyll as a proxy for phytoplankton biomass, and then we fit 2 different models for every single lake using a locally weighted regression EDM method. In the first model, the bottom-up model, the response variable was zooplankton growth rate. so the amount that the zooplankton abundance had changed from the last time point. The predictor variables were the zooplankton abundance time lags, so zooplankton abundance 1 month ago and 2 months ago, representing the within trophic level effects. We then have the chlorophyll A abundance 1 and 2 months ago, and then 2 seasonal predictors, so sinusoidal functions, to account for seasonality. The cross-trophic level effect of chlorophyll 1 month ago on zooplankton growth rate is the interaction strength that we're going to call the bottom-up effect that we're interested in and that I'm going to be focusing on for the results section. In the second model, the predictor variables were the same, but the response variable was chlorophyll growth rate. So I won't go over this model in as much detail because it's basically the same, but here we're interested in the effect of zooplankton abundance 1 month ago on chlorophyll growth rate. So this quantifies the top-down effect of zooplankton on chlorophyll. Okay, so what do you get out of an analysis like this? So here's an example from Lake Zurich, and these are the data that we're putting into these models. We have the total zooplankton abundance for every time step with a little bit of missing data in the middle, and then the chlorophyll A concentration time series as well. We can extract the top-down and bottom-up coefficients. So again, those 1 month lagged effects from the other trophic level to get a measure of the interaction strength for both bottom-up and top-down effects. So as you might expect, the bottom-up effects tend to be positive. So that means as you increase the amount of phytoplankton, there's an increase in the growth rate of zooplankton in a classical resource control scenario. And the top-down effect is generally negative, where more zooplankton results in a lower growth rate of phytoplankton. But we also see that there's a lot of variability in the strength of these effects. And you might also notice that they seem to mirror each other. So that is not something that had to happen mathematically, but just emerged from these 2 separate models. So to see which drivers were associated with variation in interaction strengths, we pooled the results across lakes and modeled the extent to which variation in both kinds of interaction strength are explained by seasonality, the trophic state index, temperature, and zooplankton species richness. 3 things that we have hypotheses from the literature about how they might influence interaction strengths. So we did this first for interaction strengths across the full range of environmental variation across all lakes. Then we're also interested in the within lake variation. And so here we centered the predictors and included a random effect of lake. And include these illustrations because they show how these across and within lake responses could potentially be different. So what actually explains variation in interaction strength? Here I've shown a plot of partial R squared or eta squared values indicating the proportionate variation in interaction strength explained by each predictor. So across all the lakes, the trophic state index explains the most variation in both top-down and bottom-up interaction strength. So the whether the lake was the super clear blue or really green impacted whether we saw the type of interaction strengths we saw. On the other hand, within lakes, none of our predictors explain very much variation in interaction strength between them, but the predictor that explained the most was just seasonality. And in both cases, there really wasn't a strong effect of temperature as we had expected, or species richness. In terms of what those relationships look like, here I plotted the mean and standard deviation of interaction strength for each lake across the trophic state index. So starting with the top-down effects, you can see that the top-down effect of zooplankton on phytoplankton in our most oligotrophic, nutrient-poor lakes is actually positive. So that means when you have more zooplankton, you end up increasing the growth rate of phytoplankton. So that might seem kind of counterintuitive at first, but actually it is consistent with literature of zooplankton nutrient recycling and excretion being an important determinant of productivity in these super nutrient-poor systems. As TSI increases into these mesotrophic lakes, where most of the previous research on the topic has been done, we see the more typical negative impact of zooplankton on phytoplankton growth rate. So top-down effect that we would expect to see, as more zooplankton are there, they're reducing the growth rate of phytoplankton. And finally, in the most oligotrophic systems, we see those interactions coming back down to zero. And so this might be explained by a greater proportion of inedible phytoplankton in eutrophic systems. A lot of them are clonal or even create toxins. Or it might just be that the zooplankton are not able to exert control over growth rates at these high concentrations. Now looking at the bottom up effects, we again see that they mirror each other, with the strongest bottom up effect occurring at mid to high trophic state index. The effect of phytoplankton abundance on zooplankton growth rate is weakest in the low nutrient systems as well. But generally, most of these values are positive, indicating that more phytoplankton increases the growth of zooplankton. Okay, so let's comparing across this really wide range of conditions between all those various lakes. But we were also interested in the within lake variation. And again, the most important predictor was just seasonality. And I pulled out 3 examples of lakes that have this really clear seasonality in interaction strengths. And we see that the magnitude of both bottom up and top down effects tend to be greatest during the warmest months. So sometime around May to August in the Northern Hemisphere and in what we would think of as winter months in New Zealand. This variation is consistent with a seasonal reset hypothesis that's been put forward, which occurs due to successional processes. So I'll describe that a little bit more. So this is on your left, an image of the plankton ecology group model that you probably have most recently seen in a textbook of some sort. And this is kind of the classic view of successional dynamics in lake ecosystems on an annual timescale. Here we have the phytoplankton and zooplankton abundances in the blue and red lines. And the shading under the blue line for phytoplankton represents the palatability or edibility of those phytoplankton, with more edible phytoplankton being these single cell, unprotected species, and the inedible phytoplankton having more spines or being more clonal. So as the ice melts in these systems, we have this rapid growth of edible phytoplankton at first, followed by an increase in the zooplankton abundance. So at the same time, in our models, we see this increasing bottom-up effect of phytoplankton on zooplankton, and the large grazers are what is appearing first. And so we see that the top-down effect of these zooplankton are also increasing at the same time. Because of this grazing pressure, there's a shift to more inedible phytoplankton, which ultimately reduces the top-down and bottom-up effect simultaneously. So I show you all this to just indicate that these species interaction strengths that we're getting from just time series of abundances do map onto known dynamics in lake systems. So that gives us a little bit more confidence in these methods and their ability to recapitulate species interactions that are well known. So to conclude this first part, so we see that species interactions or trophic level interactions are not static in time, and that different drivers are associated with this variation depending on whether we're looking across or within lakes. And so that's likely because of the greater range of environmental variation that's captured when looking across lakes. So when we're going from that hyperallergotrophic to eutrophic system, that's a lot of variation in TSI, whereas across the year within a lake, you would rarely see such a dramatic shift. We also find some results that are consistent with some existing hypotheses, like the seasonal resets and nutrient recycling, but not others. So we really expected to find a relationship with temperature, but there was no evidence of that we could find. And so the most interesting thing is that these findings emerge entirely from observational data. So of course we need theory experiments to understand why we see the relationships that we do, but these methods are able to quantify relationships and test hypotheses within whole ecosystems that would be impossible or at least difficult to do experimentally. So we propose that these methods, and building on the people who have also proposed these methods, are promising ways to explore natural variability in species interactions. And what's great about these approaches is that they're applicable to any kind of species interaction, not just consumer resources. So for example, let's say you're interested in plant-pollinator interaction, you could use time series data to look at the change in interaction strength between the two groups. So perhaps the effect of pollinators is increasing or decreasing over time. And that's something you might be able to parse out from time series. So again, this is applicable to a lot of different systems and can be applied when you have sufficient data across the relevant time scale for the interaction you're interested in. So now I'm going to switch to a second project using very similar methods and most of the same data. But instead of looking at context-dependent species interactions, we're going to look at the stability and predictability of population dynamics. So the context for our interest in stability comes from calls for ecology to be a more predictive science. And so to be a more predictive science, we also need to determine what our limits of prediction are. So determining ecological forecast horizons. I want to highlight Tanya Rogers, who is a co-author on all of this work, has a really great recent paper meta-analysis using the methods that I'm going to talk about today to determine the prevalence of chaos in ecological time series, showing that chaos is common in nature. So I highly recommend checking that one out. But for this part of the project, we were interested in time varying stability or intermittent periods of chaotic dynamics interspersed with more stable dynamics. So these intermittent periods of unstable dynamics would present barriers beyond which precise forecasts are impossible. So we're also interested in determining if we can figure out when to expect stable or unstable dynamics, and if it was related to variation in the environment, or are there certain times of year when dynamics are stable and others when they're unstable, or is there a certain scale at which they're stable, all of these kinds of questions. So what do I mean by stability? So in this cartoon, you have 2 time points in black dots. So this is from a time series of abundances. So these are 2 abundances connected to time. And if you deviate from that exact point, so now you're starting close to that original point, but off of it slightly. If you have a stable system, predictable system, all those trajectories are going to end up nearby each other at the next time point. Conversely, if you have a chaotic system or unstable system, slight deviations from the starting point are going to amplify. So they're going to result in really large differences at the next time point, which would obviously make predictions in this system really difficult. So the data that we used for this part of the project is the same data or the same data sources as our last part of the project. But here we're pulling out just individual species abundances. So time series of 1 species from each lake system. We actually also included some marine systems this time. We're also interested in how the stability changed as you looked at higher and higher trophic resolutions. So we then summed together the species abundances into functional group abundances. So here I'm showing an example where we take one copepod species for the species abundance, and we sum together all the copepods in the community to look at functional group abundance. And finally, sum together all the plankton into a trophic level abundance. We then model these using that same SMAP EDM approach, where we have the time series of the plankton, and we are modeling it as a function of time lives of itself. So the delay coordinate vector, selecting the optimal embedding dimension and time delay based on R squared from Lee-Winnowdt cross-validation. Okay, so what values are we interested in looking at? And how do we compute them? So the first thing we looked at was a measure of global stability across the entire time series. So this is a way to get a metric of, or an index of chaos. So we computed the Lyapunov exponent, or LE, as I'll be referring to it. And positive LEs indicate long-term sensitivity to the initial conditions. So therefore, they're an index of chaos. The equation's written out here and was computed for the length of the time series. So we can get the Jacobians for each time step here. And in addition to that global metric of chaos, index of chaos, we're also looked at the time varying stability. So again, we have Jacobians for each time point. And from those, we calculate an eigenvalue that is a metric of stability at each time point. So the combination of these two stability metrics tell us both the global instability, so that's the long run growth of small perturbations. And then this one tells us the time varying local instability, so the short run growth of small perturbations at a particular point in time. So here's an example of the type of results we get. So first we calculate the global stability for one population. So in this example, I'm showing data from a marine copepod in Port Erin Bay. First, we calculate the LE for the entire time series and find that the value is positive. So this indicates that this is a stable system. It's pretty close to zero, but it's positive. But when we look at the time varying instability measures, we find that there are periods of instability in the spring, late winter to early spring. So these are months along the x-axis here. And that switches over to more stable dynamics in the late summer and into fall and early winter. So using these methods, we're able to document a widespread occurrence of this intermittent chaos in natural systems, where we're alternating between periods of stability and instability. And these were really occurring on annual timescales. So you can see each of these lines across the months is a different year, and there's a relatively consistent pattern between years. So we found that our measures of stability do correspond with predictability in the system, and the interpretation of something that is globally stable but intermittent, has these periods of intermittent chaos, is a little bit hard to think about in terms of predictability. So on one hand, we're claiming that instability leads to a lack of predictability on the scale of months because of this intermittent chaos. But on the other hand, we're suggesting that the longer term predictions are possible because this is a globally stable population. So what I would suggest is the kind of safest way to think about this is that we'll probably be able to make more accurate predictions for winter and fall rather than the spring when things are less stable. And also that coarse grain predictions on an annual time scale are feasible. And that's because of seasonal entrainment. So for example, abundance will go up in spring and down in fall, though exactly by how much it might be harder to say. Again, this analysis was motivated in part by environmental change. So as the environment shifts, will we see increased or decreased stability of population dynamics? And going into it, we had these two hypotheses. So simulations have found that chaos is most prevalent at intermediate productivity, and also that community stability is known to decrease with temperature. So what did we find? Well, we didn't find very strong relationships with the environment. And in fact, we had very little evidence that stability varied with productivity at all. But there were higher LEs on average at higher latitudes where there was lower mean temperatures and more temperature variability. So this is actually the opposite of what this agrees with what we might predict. But again, these relationships are weak and mostly found once you aggregate to the functional group or trophic group level. The more consistent result that we found was that our taxonomic resolution really changed whether we found chaos or non-chaotic relationships. And so if we look at the species level time series, more than half of those were characterized as chaotic. So this is comparing the LE, so across the entire time series. Whereas when we aggregated them into functional groups and even trophic levels, the proportion that were classified as chaotic really reduced, especially at the trophic level. So this is an interesting result. We know that as you increase the taxonomic resolution, you might expect a decrease in the overall variability in biomass as you are just summing up more and more time series. But that doesn't necessarily mean that we had the prediction that one was going to be more or less stable. And so here, what this is suggesting is that it's going to be easier to predict trophic level variables rather than the individual species composition. And there's some papers that I found that in nature as well. So for example, it's been easier to predict the timing and magnitude of a spring bloom, phytoplankton bloom, rather than which species actually make up the composition of that bloom. So from this part of the project, we've shown that the prediction accuracy and sensitivity change, and therefore potentially management effectiveness, might be greater at certain times of the year. So specifically in the spring, when dynamics are more unstable, that might be a period where prediction is less accurate and the outcome of management interventions might be more variable because of the sensitivity of the trajectories to slight deviations. One thing this group talked about a little bit, I think we're pretty far away from, but could be interesting, is if we can reconstruct the dynamics enough to actually take advantage of this instability, where we can make the small perturbation in terms of management, which would make a really large change to the ecosystem. So kind of harnessing the power of that instability for our use. The other thing that we show is that prediction might be more feasible for taxonomic aggregates than for individual species. And that's useful. I would say a lot of work already does this. So for example, in lakes, I think there's more work predicting biomass of phytoplankton rather than individual phytoplankton species. Another tool that this group of collaborators has developed is another thing that can help increase the predictability of these systems is a tool called MTE-EDM, or metabolic theory of ecology, empiric dynamic modeling. And this is in a paper that was just published a couple weeks or maybe months ago now, where we use the same EDM methods, but instead of using regularly spaced time lags from calendar time, we instead use something we're calling the metabolic time step, where we basically rescale time and use shorter lags when temperatures are warm and dynamics are happening faster, and longer lags when temperatures are cooler and dynamics are happening slower. So this method, it increased the predictability of almost all of our time series, but it does assume that the speed of dynamics change, but not necessarily the form of the dynamics themselves. So I won't go further into depth on that project, but I thought I would highlight it in case someone is interested in exploring that method further. And so overall, I've shown our work using time series data and EDM to quantify species interactions and stability into these quantitative continuous variables that can be predicted in statistical models. by drivers determined by experimental and theoretical studies. So these are, I think, really awesome methods to extract a ton of information from observational data that already exists and allow us to assess how concepts where maybe we're getting different results at experiments of different scales or in theoretical work at different scales, and we can actually use these methods to test them in natural systems where all the messiness of nature is occurring. So the next steps for this work for me are expanding the scope of our work. So I'm part of a group that is developing a new database. The group that I'm working on this with was formed out of the Global Lakes Ecological Observatory Network. It's led by Steph Agari, a PhD student in Cornell, Michael Meyer, who's at USGS now, and Warren Curry, DFO in Canada. And this is a large group of people who have put together the largest freshwater plankton time series database with, as you can see, lakes that are mostly sampled at monthly or greater frequency. A lot of the results I showed you today suggest that seasonality is really important in our lake systems. And so I'm particularly excited to add more tropical lakes, more southern lakes to the analysis, and think a little bit more and have a little bit more data insight into what happens when winters become warmer and systems shift from something like a northern temperate lake to a northern hot or even tropical hot lake. We're also interested in going beyond species interactions and predictability to focus on how species influence ecosystem services or ecosystem function. So for example, I'm particularly interested in using some of the same modeling techniques on species trait to ecosystem function relationships and see if we can determine how traits scale up to influence lake functioning through time. Okay, so with that, I'd like to thank my sources of funding, my contributors to this project, in particular, Tanya Rogers, who has done a lot of the analytical work here, and many sources of lake data, and I thank you all for listening.
Thank you, Celia. That was great. We have some time for questions. So if anyone has any questions, you can feel free to write them in the Q&A. If you're online and you want to raise your hand, and I can unmute you if you want to just ask your question. Yeah. We'll maybe give everyone a minute to come up with their questions. But thank you so much, Celia. That was a really great talk.
So it's Trevor Branch here, and I'm kind of curious with this kind of big synthesis work where you're putting together data from all sorts of different people. How did you manage that big sort of synthesis part of the project of getting the data together? How hard was that?
That's a great question. I think I would classify it as surprisingly hard. I pulled up this slide because this is actually much larger scale project than the 12 lakes that we pulled together. I believe there are 246 lakes in this database that we're putting together. And so there's been years now of work coming up with ways for people to share their data in a consistent format, but then we have to actually go through each data file and actually check it and put it into the database one by one. And so it's been a really slow process. A lot of difficulties making data talk to each other and figuring out what it means when there's missing data or when there's a zero that doesn't quite make sense. All of those things have been a pretty large challenge, but I would definitely like to shout out Steph Agari and Michael Meyer for figuring it out for this larger scale project. They've got a really nice workflow for putting together this huge database. And if anyone's interested in talking to them about kind of what they've learned, I'm hoping that they'll write a paper on the process at the end, because as an outsider who's just contributing to some of the data cleaning, I think it's gone really smoothly.
Thank you.
Great. It looks like Sam has a question. So Sam, I'm going to let you talk. Hopefully that works.
Hey, Sam.
Sam, are you there? Oh,
Maybe not.
it says you're not muted, but we can't hear you.
Maybe if he just types it out.
You might need to type your question out, Sam. Sorry. Well, does anyone else have any questions? All right, well, it looks like there might not be any questions. So thank you so much, Celia. And thank everyone for coming. And we'll see you next week for another quantitative seminar.
All right.
Thanks.
Thanks, everyone.
Thank you, Celia.
Oh, we got it. Can you read it, or would you like me to read it? I think everyone
Yeah.
can see it.
Oh, everyone can see it? Okay, so
Yeah.
let me read it. So EDM approaches are very cool, I agree. The limits of time lag in real ecosystems.
Oh, here.
Yeah. Yeah, so the question is about kind of the limits of the time lag embedding approach in real ecosystems, and especially in systems where the data points are at a less frequent timescale and substantial uncertainty associated with them. Yeah, I mean, I think like there's real limits to what we can do based on that, especially if good to see you too. Especially if the timescale of the observations are not matching up with the timescale of the interactions that you're interested in. That's been kind of our biggest issue with getting data at a frequent enough timescale. So, you know, even a month-long time step between points here, you're capturing not only the direct impacts or direct interactions between these trophic levels, you're probably also capturing a lot of indirect interactions. And so how those kind of add up to equate to the interaction strength over the course of the month can be a little bit challenging. And so that can be, you know, even more of a problem if you're only looking at annual or biannual timescales. Of course, if your dynamics are happening over those timescales, perhaps you're interested in the interaction between two whale species or something, and then that could be appropriate. Hopefully that's what you're asking.
Okay, great. I'm glad we got that question in. That was very interesting. If there are no other questions, then I think we will end the seminar there.
Right. Thanks, everyone.
Yeah, thanks so much, Celia. Hope the concert's nice out there.
Thank you.
Bye.